Good morning, my dear students. Myself, Dr. Rathod Lal Singh, Assistant Professor in the Department of Agricultural Entomology at School of Agriculture, SR University. In this session, we are going to discuss about one of the most important biopesticide, that is EPN, entomopathogenic nematodes. Entomopathogenic nematodes. These are the soil-inhabiting nematodes or organisms. This nematode causes diseases in insects. That's why we are calling them as entomopathogens. This nematode lives inside the host body. That's why they are considered as endoparasitic or endoparasitic nematodes. The nematodes, which belong to the steinernema family and Heterorhabditidae family, are most useful as the biocontrol agent in the various insect pest management. We will see the different species and host insects in coming slides. Entomopathogenic nematode has 6 life stages. They are juvenile 1, juvenile 2, juvenile 3, juvenile 4, juvenile 5, and juvenile 6. In general, that is, in case of plant parasitic nematodes, juvenile 2 stage is infective or effective 1, but in case of entomopathogenic nematodes, juvenile 3 stage, that is J3 stage, is infective or effective 1. This entomopathogenic nematode have symbiotic relationship with the bacteria, and this symbiotic relationship is compulsory in order to kill insect pest. If there is no symbiotic relationship with bacterium, then these nematodes are ineffective or useless. See here the example for nematode-bacterium complex. Steinernema nematode, which is associated with the Xenorhabdus bacteria. Heterorhabditis nematode, which is associated with the Photorhabdus bacteria. Then, what is the benefit for nematode from this symbiotic association from the bacteria? Bacteria kills the insect host, create a suitable environment, break down the host tissues, and provide the food source for the nematode. These are the benefit to the nematode from the bacteria. Then what is the benefit for the bacterium from the nematode in this symbiotic association? This nematode helps the bacteria in the penetration of this bacteria into the host hemocell, and this nematode protect the bacteria from the external environment as well as from the host antibacterial proteins. Otherwise, this antibacterial protein neutralize that bacteria. Like that, in this symbiotic association, bacterium is benefited from nematode and nematode is benefited from the bacteria. See here the scientific classification of steinernema nematode. It is belong to the kingdom Animalia, phylum Nematoda, class Chromadoria, order Rhabditida, family Steinernemachidae, genus Steinernema. See the various species under Steinernema nematode and their host. Steinernema carpocapsae. Steinernema carpocapsae, it is effective against corn earworm, that is Helicoverpa zea, fall armyworm, codling moth, European corn borer, and various beetles and other soil-dwelling insects. Next species is steinernema feltiae, which is effective against fungus gnat, root weevil, cutworms, carrot weevils, and various soil-dwelling pests. Next species is Steinernema glazeri. It is effective against a sugar beet root maggot, wireworms, corn rootworm larvae, various beetle larvae, and other soil-dwelling insects. Next species is Steinernema krasi. It is effective against weevils, fungus gnat, and various soil-dwelling insect pests. Next species is Steinernema longicauda, which is effective against wireworm, beetle larvae, and various soil-dwelling insects. Next, see the scientific classification of Heterorhabditis nematode. This Heterorhabditis nematode belongs to the kingdom Animalia, phylum Nematoda, class Chromadoria, order Rhabdicida, family Heterorhabditidae, and genus Heterorhabditis. See the species under Heterorhabditis nematode. Heterorhabditis bacteriophora. It is effective against corn or rootworm larvae, beetle larvae, weevils, cutworms, and various soil-dwelling pests. Heterorhabditis indica, it is effective against sugarcane borer, root weevils, fungus gnats, various soil-dwelling insects. Heterorhabditis marillatus, it is effective against root weevils, beetle larvae, wireworms, various soil-dwelling pests. Next, Heterorhabditis downsii. Heterorhabditis downsii, it is effective against beetle larvae, cutworms, various soil-dwelling insects. Heterorhabditis safricana, it is effective against root weevils, various beetle larvae, and soil-dwelling insects. Next, we are going to see about mode of action of entomopathogenic nematode in brief. This entomopathogenic nematode actively forays and seek the host in the soil. Once it find its host, it penetrate into it through the natural openings such as anus, mouth, spiracles, or any other sharp part of the body, and cause infection by releasing bacteria. Inside this host, the bacterial pathogen release toxins and other toxic enzymes that cause pathogenesis, that cause diseases in the host insect and eventually kills them. On this dead and decaying host, the nematode feed, grows, reproduce, and produce progeny, and this progeny will disperse for attack to the next new host. See here in detail the mode of action of entomopathogenic nematodes, foraging and host seeking. Entomopathogenic nematodes actively search for the insect host in the soil. They are attracted to the chemical signals such as carbon dioxide and other compound emitted by their target host. These chemicals guides these nematodes towards their potential host. Next step in the mode of action is the penetration and infection. Upon locating a host, entomopathogenic nematode enter into the insect body or host body through the natural openings like mouth, spiracles, or soft areas of the cuticle. Once inside, they release symbiotic bacteria, either Xenorhabdus or Photorhabdus. It's depend on the nematode species. Bacterial pathogenesis. The bacteria inside the host develop and multiply rapidly, secrete toxic enzymes and toxic material that break down the host tissues and suppress host immune system, leading to the death of the host. Nematode feeding, reproduction, and dispersal. Entomopathogenic nematodes feed on both the bacteria and the decaying host tissues. It feeds on the bacteria to carry its infectious nature, infectious nature to attack the next host. And decaying host tissue, it feeds for its development and reproduction. As these nematodes consume these sources, they grow and reproduce. The nematode produces new infective juveniles which exit the dead host and search for the new targets. Different formulation of entomopathogenic nematodes. Entomopathogenic nematode available in granular form, liquid suspension, wettable powder form, pellet form, and capsule form. In granular form, entomopathogenic nematode is mixed with a granular carrier materials. In liquid suspension, entomopathogenic nematode suspended in water or any other liquids. In wettable powder form, EPNs formulated as a powder. It's to be mixed with the water. In pellet form, EPNs are produced in pellet or crumble form. In capsulated form, EPNs are developed into a capsule, that is biodegradable capsule for slow release of the nematodes. Granular form is applied as are applied for soil-dwelling pest. Liquid suspension, it is suitable for soil and foliar application. A wettable powder also suitable for soil and foliar application. Pellet form, it is also useful for plant and soil application. Encapsulated form also useful for plant, that is whole application and soil application. See here the typical dose, it is ranges from 1 to 5 billion nematode per acre, and it will be vary with nematode to nematode species and their age and stage. See here commercial formulation given here. Not grub stake high, horticultural scan mask, Nemashield, nemasys and their doses for hectare is given. That is 25 million infective juveniles can be cover 1 to 2 hectare, or we have to mix in 200 to 500 liter of water to spray 1 hectare area. See here one experimental example is given for the management of root grub in erica nut. For management of root grub in erica nut, we have to apply steinernema carpocapsae in a liquid suspension at a rate of 1.5 billion infective juveniles per hectare. The first applications should be made during June-July with the onset of the monsoon. The second application should be done during September-October. Along with the application of this entomopathogenic nematode, apply imidacloprid. Imidacloprid 17.8 SL at 0.5 ml per 2 liter of water per farm. It acts as synergistic with the entomopathogenic nematode and enhances the control of root grubs. One important noting point. During nematode application, regularly irrigate the field to maintain optimum soil moisture. Why? Because it is a very must or which is crucial for survival, effective establishment of nematode and their movement from one place to the another place. Environmental persistence. In absence of host, that is outside of host infection, EPN do not undergo dies. Instead of this, EPN can survive the soil by entering a dormant state. They withstand various environmental conditions and remain viable, ready to infect a new host when the condition becomes favorable for them. See here the eco-friendly powder form entomopathogenic nematode formulation. It is recommended at the rate of 1 into 10 to the power of 8 infective juveniles per acre, that is 2 kg powder formulation per acre. This is the gel formulation of entomopathogenic nematode. In the final slide, what are the symptoms of entomopathogenic nematode in host insects? The symptoms includes cessation of feeding, increased hemolymph pH, vomiting, diarrhea, sluggishness in host insect. We can also observe general paralysis and septicemia, that is blood poison occur in 24 to 48 hours, resulting in the death of the insect. In particular to the steinernema nematode infected larvae, they turn creamy or dark brown in color. At the same time, heterorhabditis nematode infected larvae, they turn reddish or purplish in color, and they also shows sticky nature of the cadaver. Thank you. In the next session, we are going to discuss about mass multiplication of entomopathogenic nematodes. Good morning. Welcome you all. Now we are going to discuss about steps by steps process for field isolation, recovery or extraction, mass production and field application of entomopathogenic nematodes. The first step is field isolation of entomopathogenic nematodes. So for this, gather soil from the different part of the field and place this collected soil into the plastic container. Into this plastic container, introduce 3 to 5 waxworm as the bait, since they are highly susceptible to entomopathogenic nematodes. Then after, close and keep the container in the dark room at 20 to 25 degrees Celsius and check the worms for the sign of EPN infection over 3 days. The infected waxworm will change their color and they die. If no infection is observed, collect the new soil sample and repeat the same process until the infection is detected. If there is infection, note the color change in the infected waxworm to identify the EPN species. For example, if the waxworm is infected with heterorhabditis bacteriophora, it develops brick red or purple color. If it is attacked by heterorhabditis migidis, it develops orange color. If it is attacked by steinernema glazeri, it's develop cream or brown color. So like that we can isolate EPN from the field. Next, recovery of entomopathogenic nematodes from the infected cadavers of the waxworm by using white traps. So for this, first rinse the dead cadaver of the waxworm with the sterile water to clean off soil and dirt. Next, cut a Wattman filter paper to fit a convex watch glass and place this watch glass in the petri dish. Moisten the filter paper with the sterile water and place it over the convex watch glass which is already placed in the large petri dish plate. Next, place the cadaver and the filter paper with the little distance between them for easy movement of emerging juveniles. In this petri plate, pour 20 ml of sterile water to keep the filter paper moist. Over 7 days or over 7 to 10 days, EPN will move into the water and gives the appearance of cloudy. The water will give the appearance of cloudiness. It indicates the nematode emerged from the waxworm larvae. Like that, we can recover entomopathogenic nematode from the cadavers of the waxworm. Now, we can use this cloudy water, cloudy entomopathogenic infected water for the mass multiplication of the entomopathogenic nematode. See in the next slide, mass production of entomopathogenic nematodes. Pour 2 tablespoons of EPN infested water over 250 waxworm larvae in the container. So this container contains 250 waxworm larvae over which we have to pour 2 tablespoons of entomopathogenic nematode infested water. Keep this container in a dark, moist environment at room temperature. After 2 weeks, EPN will emerge. Yellow moss on the lid and wall of the container will indicate the emergence of entomopathogenic nematodes. Now collect them by sieving the content and washing with the distilled water. So we can collect in this tub or container. Next, field application of entomopathogenic nematodes. Mix this entomopathogenic water solution in the spray tank and spray entomopathogenic nematode on foliage or the soil late in the afternoon or around the sunset to avoid UV damage to the nematode. Take 2 points in consideration, that is, keep the soil moist to help entomopathogenic nematode establishments and their movement. Second one is till or move the soil for easy spread of the EPNs throughout the field. So, see here, each waxworm larvae can bear approximately 1 lakh entomopathogenic nematode. Therefore, 250 waxworm can bear 25 million entomopathogenic nematodes. But we need 100 to 300 million EPNs per acre. Therefore, 4 cups of EPN infested water, each containing 250 waxworms, are needed to apply the recommended dose of 100 million EPNs for the acres. Thank you. Now we will see objective type questions related with the entomopathogenic nematodes. The insecticidal property of nematode was discovered by Dutke and Howe. Dutke and Howe. The infective juvenile nematode is, which instar is infective juvenile of the nematode, that is third instar is infective juvenile. Steinernema and Heterorhabdidae are associated with the bacteria, whereas nematode belong to the Myrmidhidae are the obligate parasites. They do not depend on the bacteria for killing the host. They kill they themselves. In steinernema and heterorhabdites, bacteria are stored in the intestinal lumen of the nematode, and infestation result in the formation of bacterial soup or bacteremia. Bacterial soup refers to the condition within an infected host where the accumulation of bacteria and host tissue fluid create a thick, viscous mixture. Myrmidhidae cause morphological abnormalities and sterility. An example is hexamermis nematode. See, this is the hexamermis nematode. Steinernema neoplectana or Steinernema glacier is the first entomopathogenic nematode applied in the field to the turf against the Japanese beetle Popilla japonica. Steinernema glacier is the first nematode applied in the field to the turf against Japanese beetle Popilla japonica. DD-136 formulation is the mixture of nematode Steinernema carpocapsae and the bacteria Achromobacter nematophilus. So DD formulation is the mixture of nematode Steinernema carpocapsae and the bacteria Achromobacter nematophilus, and it is applied as an aqueous spray for the control of codling moth, that is Cydia pomonella. This is the Cydia pomonella. For this pest management, we can use DD-136. Steinernema nematode release Xenorhabdus bacteria and Heterorhabditis nematode release Photorhabdus bacteria. Therefore, this nematode act like a small syringe to inject the bacteria into the host. In Steinernema, the symbiotic bacteria is from the genus Xenorhabdus, specifically Xenorhabdus nematophilus and Xenorhabdus bovini, but this bacteria do not flourish. But in case of Heterorhabdidae, the symbiotic bacteria is from the genus Photorhabdus, specifically Photorhabdus luminescens. This bacteria fluoresces glow in the dark, making the infected larvae easy to detect. Steinernema-nematode infected larvae turn into the creamy or dark brown in color, whereas heterorhabdid nematode infected larvae turn reddish or purplish in color. See here the nematode and their association with the pest. Tetradonema pelicans. It is effective against cearid flies, that is fungus gnat. That is the pest of the cultivable mushroom. Second nematode, Romanomyrmex culicivorax. Romanomyrmex culicivorax is effective against mosquitoes. Steinernema feltiae, it is effective against soil pests and termites. Rhabditis species, it is effective against Holotricha serrata, a type of beetle pest. Based on their searching strategies, entomopathogenic nematodes are two types, that is ambusers and cruisers. Ambusers, they remain near the soil surface and wait for the mobile host to come close. But cruisers, they actively search for the host within the soil and are effective against less motile insect. Example for ambuser is steinernema carpocapsae. Example for cruisers is steinernema glazeri, heterorhabditis bacteriophora. Ambusers target soil-dwelling and surface pests; cruisers target less motile soil insects. Based on application and targeting strategies, entomopathogenic nematodes are green commandos and soil commandos. Green commandos, entomopathogenic nematode used for foliar application. Soil commandos, entomopathogenic nematode used for the soil application. Example is steinernema species. Soil commandos are heterorhabditis species. Application: green commandos apply to the leaves and above ground parts of the plant, applied to the soil. What do you mean by entomogenous nematode and entomophagous nematode? Entomogenous nematode, they are generated from the nematode. They feed and multiply on the host. But in case of entomophagous nematode, that nematode only feed on the host without reproducing inside it. Example for entomogenous, they are generated from the insect, that's why we are calling them as entomogenous. Example, steinernema species, heterorhabditis species. Entomophagous, they feed on the insect host, that's why we are calling them as entomophagous rhabditid species. Nematodes are mass cultured in vivo in the lab by using lepidopteran larvae like Galeria melonella, that is waxworm larvae, Quercera cephalonica, rice moth larvae, and Spodoptera lichura. The National Center for Integrated Pest Management is situated in New Delhi. Thank you. Thank you all.